Thank you. Thank you very much. And first, I wanted to thank David and all of his colleagues for organizing a great meeting. This is my first Li-Fraumeni meeting, and I was very moved by some of your stories this morning. And I hope that the little bit of research that I do might eventually impact how we treat patients. So I'm going to be talking about p53 missense mutations and how they contribute to additional activities that lead to very invasive cancers. So first, this is a reiteration of slides that you've already seen, but it's basically the kinds of mutations that we see in Li-Fraumeni patients. And the kinds of mutations are, in general, missense mutations in the DNA binding domain. So this is very different from not having p53. These are mutations that still result in the production of a protein, but this protein is no longer functional. There are hotspot mutations. I'm going to be talking about this 175 mutation, and just the fact that these patients have a p53 mutant protein, and the fact that these mutations are very common, led many in the field to think that there's really an advantage for a tumor cell to have these p53 mutations. And so my lab and that of Tyler Jacks made the first Li-Fraumeni mice. So we mimicked that 175 mutation, which is 172 in the mouse. And basically the reason I use the mouse as a model, it's a vertebrate, it's a mammal, but also because we can manipulate the genome of the mouse and make a single point mutation similar to what we see in human patients. So this is just one slide from a lot of work that shows that many of these primary tumors express this mutant p53 protein. And in comparison to mice that have no p53, these tumors are very aggressive. So here you can see a very large metastasis to the liver, and then here is a small metastasis to the brain, and this is stained with the p53 antibody. And I think from this slide you can see that this metastasis is in a sea of cells that don't have the mutant p53 protein. So some of the studies that we did to characterize the mutant p53 stability in normal cells showed that this is normally not a very stable protein. So when it's not a stable protein, it actually mimics not having p53. And so the important question that we're trying to address is what stabilizes that mutant p53 in tumors. We know that many of the signals that stabilize wild-type p53 also stabilize mutant p53. And so this is just a Western blot that shows some of those activities. So here is our animal model that is homozygous for this p53 missense mutation. Again, this protein is not a very stable protein in vivo, but when you irradiate this mice DNA damaging agent, ionizing radiation, you have increased stability of this mutant protein. In addition, we generated crosses with other mice in which we can delete this MDM2 gene. It's an E3 ubiquitin ligase that targets p53 for degradation, and in the absence of this protein, in the absence of MDM2, this mutant protein is also very stable. And so we looked at these mice to ask what happens when we artificially stabilize that mutant protein by deleting p53, and it really is a much more aggressive cancer. So this shows just the fact that by deleting MDM2, we do stabilize that p53 protein in many tissues, but not all. And then this is the slide that shows the aggressive phenotype of these cancers. So here in the black is the survival of mice that have this missense mutation, and it's 100% penetrant, and all of these mice are dead by about a little less than a year. In the absence of MDM2, as I just showed you, we stabilized that mutant p53 protein, and that led to an increased cancer risk. These mice died much sooner, and these mice had a high metastatic burden. And so what we know then is that in normal individuals, this is not a mutant stable protein, but in certain situations when that protein becomes very stable, it can be very deleterious. So we and others are trying to understand how those p53 mutations give rise to more aggressive cancers. Varda, Rotter, and Carol Privis showed that transcriptional activity, that transcriptional activation domain of mutant P53 was critical in driving new transcriptional programs. And so we focused on the osteosarcomas because that's the most common cancer that we see in these patients, or one of the most common ones. And what we have here is a comparison of tumors that have no P53 and very little metastases to tumors that have this P53 mutation and are highly metastatic. And you can see there are about 200 gene differences between these two kinds of osteosarcomas. And I'm going to just give you a brief description of this one gene that we targeted. It's called the phospholipase 2G16. I'll call it a phospholipase from now on. And I'll tell you why we picked this one gene to study. But here on the right, what I've shown is a validation study. You can see here in this sample, these mice have no P53 and have tumors, but their expression of this phospholipase is very low. And with the mutant P53, you can see very high levels of this phospholipase. So this phospholipase was of interest to us because it's involved in lipid metabolism. So it cleaves this covalent bond in phosphatidylcholine, which releases a free fatty acid and generates this lysophosphatidylcholine, which is then converted to lysophosphatidic acid. Believe me, I had to open my old biochemistry book to remember all of these pathways that I had studied years ago. But the reason this was of interest is because LPA is a known facilitator of invasion and migration in people. So we've manipulated the expression of this phospholipase in tissue culture, and what we've done here is taken tumor cell lines that have a mutant P53, and we down-modulate this phospholipase and ask, how does eliminating that phospholipase affect migration and invasion? And you can see here in two different cell lines, deleting or down-modulating this phospholipase decreases the migration and the invasion potential. And this here on the right is an in vivo system where we ask, what is the capacity of these cells to induce metastasis in syngeneic mice? And you can see here the control mutant P53 with high levels of this phospholipase gives you a very high incidence of metastasis, but when we down-modulate that phospholipase, we decrease the metastatic phenotype. So this phospholipase is downstream of this mutant P53, and it is contributing to this metastatic phenotype. We're trying to understand how this mutant P53 regulates this phospholipase. And so here on the left, we did what we call a chip experiment and identified this mutant P53 binding to this phospholipase at several sites. And so when we down-modulate that, back up for just a second, that mutant P53 correlated with a binding site for this ETS2 transcription factor. And when we down-modulate that ETS2 transcription factor, you can see we see a decrease in the phospholipase activity. And that doesn't happen with the related transcription factor. So what we think is happening then is that this mutant P53 is interacting with this ETS2 protein to drive expression of this phospholipase, which then leads to these migratory and invasive phenotypes in tissue culture. So I guess this is that slide. Basically, that mutant P53 protein still has a very potent transcriptional activation domain, which can bring in the rest of the transcriptional machinery and drive expression of this phospholipase. But I don't want to leave you with the impression that this is the only gene that mutant P53 regulates. But what I would like to focus on for the next few slides is this ETS2 protein and how critical this ETS2 protein is in driving metastatic phenotype. But first I have to introduce a new mouse, and that's the mouse where everything in the whole mouse is wild type for P53, and we can make the mutation in a specific cell type. And so we chose to make the mutation just in osteoblasts to get osteosarcomas. And the reason this is so critical is because in our animal models, we get all kinds of tumors. We get lymphomas, we get sarcomas, we get some in different backgrounds, we'll get some breast carcinomas. And it's very difficult to follow when these mice are going to get tumors and when they're going to get metastasis. So we were hoping that by concentrating just on osteosarcomas, we could develop a very pretty metastatic model to understand the disease better. So this is the allele that we've made, and basically it's an allele where we have expression of wild-type p53 because of the presence of this wild-type cDNA sequence. But then in a Cre-mediated fashion, we can delete these wild-type p53 sequences and now actually make a mutant p53 locus identical to the one that I've just described in the whole mouse. And so then this leads to the expression of this mutant p53 protein. So we first did a lot of work to make sure that this mouse was really a normal mouse and it was wild type and didn't have a tumor phenotype. So I'm just going to show you a couple of slides that tell you how normal these mice really are. So we call these WM alleles because they're wild type to start with, and then we can make them mutant at will. This is just an assay in which we've irradiated cells and we compared the wild type to the two different mutations that we made. So now we've made the 172 mutation, and then we've made this R245 mutation, which is equivalent to the 248 mutation in humans. And you can see that irradiation stabilizes that wild type p53. And here you see expression of different p53 targets, and you see no difference between these two bars, indicating that this WM allele is really doing what wild-type p53 should be doing. We've looked at p53 responses. We know that wild-type p53 can induce apoptosis, and here there's no difference between the ability of these WM alleles to induce apoptosis. On this slide, this is a cell cycle arrest assay, and again, there's no difference in the ability of this WM allele to initiate an arrest program. And this is like the golden, the assay that took 3 years almost to generate, but basically what we're comparing here is the tumor phenotype of the mouse that's normal for p53 and the two mice that have these WM alleles. And I don't have to point out a color here. All of these are identical. They're all overlapping survival curves. So what this says is that these alleles are as normal as a wild-type mouse. So given all of these data now, we can make a specific point mutation in just a single cell type. So what we chose to do is to make a p53 mutation in osteoblasts, which is the origin of osteosarcomas. So again, this is the 172 allele, and we use this ostrus Cre promoter to delete the normal p53 sequences and allow expression of just this mutant p53 protein. In addition, the beauty of this Cre system is that you can also label your cells a certain color fluorescence. And in this case, we use this MTMG allele. So normally the cells are going to be red fluorescent, but when the mutant p53 protein is expressed, these cells are going to be green. And we got these beautiful osteosarcomas. So here in this first image, I have a X-ray of a mouse that has this very large osteosarcoma here. As I indicated, this osteosarcoma is going to be green. So here you can see all these green dots in the lung. Here's an expansion of some of those metastatic nodules in the lung, and then another one here in the liver. And so these mice develop 100% penetrant osteosarcoma, and they develop a lot of metastases. Here are the data for the metastases now, comparing this model in which we're making a p53 mutation only in the osteoblasts versus a model where we're deleting p53 only in osteoblasts. And you can see there's a clear increase in the metastatic capacity of the cells that express this mutant p53. I don't want to leave you with the impression that not having p53 isn't bad. It is bad, but having a mutant is actually worse. In addition, what I'd like just to sort of emphasize is that this is going to be a beautiful model for us to begin to understand the steps from the development of that first osteosarcoma to the development of these metastatic nodules. And this is an in vivo system, so it's not an artificial system in tissue culture. It's actually a physiologically relevant model system. So we think it's a great model for us to understand the stepwise progression of this metastatic phenotype. Okay. I had told you earlier that we thought ETS2 was a critical regulator of this metastatic phenotype. So what we've done here is an incredibly difficult experiment, because now we've taken mice that have this P53 mutation, and we took out the other wild type allele, so it only expresses mutant P53. And then we also took out this ETS2 gene with this osteoscrete promoter. So what we're doing is we're generating a model in which we have this P53 mutation, and on top of that, we're taking out this ETS2 protein. And here's the data on the right. So here's the mouse that has a mutant P53 protein, and this was we had about 50% metastatic disease. And when we take out ETS2, you can see that that drops to about 10%. So ETS2 in vivo is a critical driver of this metastatic phenotype. We've done some RNA analysis, and this is just an analysis. So ETS2 is the transcription factor. Mutant P53 is interacting with ETS2, and it's driving a transcriptional program. One of the things that we noticed was that there was ETS2 was driving expression of these RNAs that we call SNO RNAs. I don't really know what they do yet, but there's a whole series of these SNO RNAs that were identified in our samples. So here in red are, well, these are, across here are, I think, about 25 different SNO RNAs. Here in red are the mice that have our mutant P53, and you can see that they express these RNAs at very high levels. In the mice that have no P53, here in blue, they don't express these SNO RNAs at very high levels. And in black are the mice that have a mutant P53, where we've taken away ETS2, and you can see that those samples, those mice also express very low levels of these SNO RNAs. So my last slide simply is trying to put all this in perspective. I think the data are very clear now that ETS2 is a driver of a metastatic phenotype. So I've talked mostly about this one P53 missense mutation, and there's really two things that are happening here. One is that it is a loss of function. It can't activate transcriptional targets. It can't induce cell death when it needs to. And so it's like not having P53. But in situations where that P53 becomes stable, then it interacts with this ETS2 transcription factor and drives this differential expression of mRNAs. The phospholipase 2G16 was one of them, and now we have this whole group of SNO RNAs that we know are regulated by a mutant P53 and ETS2, specifically in osteosarcomas. So I'm hoping that this model system will help us understand a little bit more about the progression of the disease. I think a critical question that we still don't have a very good handle on is what leads to the stability of that mutant P53 protein, because it's that stable mutant P53 protein that's driving a metastatic disease. So I'll end there. This is my lab. It's a great group. I'm very grateful to them that they take on these very long-term projects. But perfect timing, thank you. Yun Zhang is the person who made this WM allele, and she's doing some other beautiful studies with breast cancer. She got a lot of help from Shunbin Shang, and then Rasul did the studies with the osteosarcomas. So I'll end there, and I'll take questions. Thank you.
Perfect, we have time for multiple questions. Carol.
I'll repeat the question.
Yeah, this one works. Does mutant P53 co-bind with X at the SNO RNA?
Great question. We haven't done any of those
And do you know
experiments.
what drives SNO-A? It's not POL2, is it? These SNO RNAs.
I don't remember. We looked.
Pole 1.
It's pole 1? Thank you,
So then
Ernie.
that means
Yes,
X.
and so there are, all of these SNO RNAs do have an ETS2 binding site in the promoter.
They do.
They do. So we think that that's what's happening. We want to do chips with both mutant and ETS2 at the promoter to actually ask that question. The problem is that I want to do this in vivo. I want to do this in the osteosarcomas. I don't want to do it in tissue culture cells. And these are very bony tumors, and so we're trying to get the assay. But yeah, we're trying to get the assays up and running. Arnie.
There's an interesting coincidence of facts that all the SNO RNAs are in the nucleolus.
Correct.
And they probably play a big role in ribosome biogenesis.
Correct.
And so does MDM2 surveil ribosome
Correct.
biogenesis. So I wonder if there isn't some
yeah.
connection here between the players, so to speak, that now SNO has come in. I know that methylation of RNA is a big deal in ribosome biogenesis, as well as the assembly of ribosomal proteins, which is ultimately the key to feedback on P53 and MDM2. So it seems to me there's some connection that...
No, you're absolutely right. These are nucleolar proteins, and there's that literature in the P53 field that contributes to stress and to elevation of P53 levels. So yeah, we're going to have to try to figure out how those signals intercept. Mm-hmm. Yes.
So H2 is an important transcription factor in the regulation of epigenetic driver genes such as ML1, MLL1,
Yes.
MLL4.
Mm-hmm.
Is there, and you probably know the work of Shelley Berger,
Yes.
who has suggested that gain-of-function mutant could co-recruit the expression of such genes. Do you have expression patterns in
We haven't looked.
gain of function that could be similar to this? That could
No, you're absolutely right. There's a role for ETS2 in the regulation of chromatin modifications, and we're slowly transitioning to an epigenetic lab, as much as I don't like to.
We have time for one more. Anyone? All right. Well,
Okay, great. Thank you.